Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Before we begin, a note from our sponsor. I'm Richard Jacobs, Executive Director of the nonprofit Finding Genius Foundation and host of the Finding Genius Podcast. In late 2016, I was rear-ended at 65 miles an hour by a truck on the highway, which sent me off-road into a ditch. The impact of the collision gave me a concussion and other injuries. At the hospital, a CT scan showed that I had thyroid nodules, which turned out to be cancer. It was then, when I had a biopsy of my neck, that I realized even if I was a millionaire, I wouldn't want a second or a third biopsy due to the pain and the invasiveness of it. And appointments at that time for thyroid experts were 3 to 6 months out. And I was worried about dying now, even if that was irrational. So because of this, I've decided to raise money to conduct a literature review on steroids, on the causes of anxiety and depression, a condition that affects well over 50 million people in the United States and hundreds of millions worldwide. Our goal is to create a codex, a guide that reveals all possible treatments for anxiety and depression for people that live with the condition, or for loved ones that have it, as my wife and my son do. To find out more about our fundraiser, visit findinggeniusfoundation.org and click on current initiatives. And now, to our guest.
Hello, this is Richard Jacobs with the Finding Genius Podcast, now part of the Finding Genius Foundation. I have Dr. Emeran Mayer. He's the author of a new book called The Gut-Immune Connection, How Understanding Why We're Sick Can Help Us Regain Our Health. I spoke to Emeran a couple of years ago. He's very knowledgeable about the microbiome, and I'm glad to have him back. So, Emeran, thank you for coming back. I really appreciate it.
Yeah, Rich, thanks for having me on the show. It's a pleasure.
Yeah. Well, tell me, how did you get involved in medicine and research, and what were you doing before this? What got you interested, especially in the microbiome?
What got me interested in medicine? I have to be honest. I'm not somebody who knew from first grade on that, or my parents didn't think that I would go into medicine. I struggled throughout, end of high school, college, to really make a decision what professional path I wanted to take. And that included everything from what was hot at the time: sociology, political sciences, psychology, and medicine. But medicine was definitely not one of the top choices. And then really science has always interested me as a kid, not necessarily biological sciences. But then, based on my grades, the GPA I had, everybody was saying, oh, you have to go to medical school. And really in the last minute, I decided to do that, last minute that I could apply. And somehow I think these different interests have sort of stayed in my mind, in the background of my mind. I'm not somebody who has exclusively an interest in hard science and in medicine, even though I've greatly enjoyed my career, both helping patients and being at the cutting edge of research in the brain-gut field for pretty much all my professional career. And the decision to pick brain-gut interactions, so initially was just brain-gut, then later became brain-gut microbiome. It was there from the beginning, I think, because it sort of allowed me to incorporate my psychological interests, with medicine. And I've done this for almost like I would say 40 years, going through very different stages of research, from studying isolated cells in the test tube, recording from isolated nerve cells, to doing studies in mouse models, to then switching almost completely to human studies. And about 7 years ago, when first microbiome science, the first spectacular findings were published that the microbes could actually influence behavior and the brain, I was initially skeptic, and then incorporate that later in our research center in my own research program. And, that has become sort of a major focus. So now the brain-gut microbiome axis is sort of the topic that I'm most interested in.
Right. Okay. Well, tell me about your new book. What new information did you put in there? Why did you write this latest one?
Yeah, so this was something, certainly being involved, being immersed in the science of this, and also now, being faced with a tremendous, after my first book, most of my patients came to my office with the book in hand and wanted to have it signed and want to have it explained, or saying that some of the patient stories I had in that book, that was them. It was so similar to what they had experienced. So it was so I thought it's a good time 5 years later to write another book, to update a lot of things that have happened in the research field. When the first one was written, there were a few animal studies that we based most of this concept on. And my agent at the time, reading the first draft, said, "This is ridiculous. Nobody's interested in mouse studies. The people that you want to target are interested in human studies." And that was hard at the time, quite honestly. So now I think we have a lot more studies. So I felt for the first time I could really write an authoritative book discussing the latest studies in humans and what this means for human diseases, brain diseases, and others as well. And so that was one. The other one was, while writing the book, this was definitely a personal growth process that, the pandemic happened. There was a lot of discussion about this epidemic of chronic non-infectious diseases that is going on with metabolic diseases, and so not just in the brain, but really affecting every organ system in the body, that are now being linked in some ways to the interactions of the microbes with the immune system. And that sort of became really the main focus of this book, So the first time it was all microbiome brain. This time it's microbiome, it's diet, microbiome, immune system, and other organ systems.
Okay. So what when people talk about the gut-brain axis, what does that look like? What's it mediated by? Like, how does the communication happen between the gut and the brain? Let's start with that.
So the gut-brain axis is a bidirectional. It's actually, I use now the term brain-gut microbiome system, because it is not just an axis. It's not a linear communication system, but it's really a what we call, like in systems biology, something nodes that are interconnected by bidirectional loops and feedback loops. So it's much more complicated than an axis. What it is we have known for quite some time that the brain can send signals to the gut and change pretty much the entire repertoire of gut functions. The gut has its own nervous system, the enteric nervous system, or little brain of the gut, which takes care of pretty much in an autonomous way of all gut functions and adaptations to diet and what we put in it. But it's dependent on influence from the brain to inform it about emotional states of the brain, about perturbations that happen in the rest of the body outside the gut, and also stressors that occur from the environment. So the gut can run on its own, and there have been experiments, with mouse intestine or guinea pig intestine, that a piece of isolated intestine in a test tube can pretty much do the same peristaltic contractions as an intact gut, but it cannot respond to all these other influences that I just mentioned. So the brain, in response to, I prefer the term perturbations of homeostasis to stress. So anything that either threatens or actually does perturb the homeostatic action of the body.
Before we continue, I've been personally funding the Finding Genius podcast for 4.5 years now, which has led to 2,700 plus interviews of clinicians, researchers, scientists, CEOs and other amazing people who are working to advance science and improve our lives and our world. Even though this podcast gets 100,000 plus downloads a month, we need your help to reach hundreds of thousands more worldwide. Please visit FindingGeniusPodcast.com and click on Support Us. We have 3 levels of membership from $10 to $49 a month, including perks such as the ability to see ahead in our interview calendar and ask questions of upcoming guests, transcripts of podcasts you're interested in, the ability to request specific topics or guests, and more. Visit FindingGeniusPodcast.com and click Support Us today. Now back to the show.
It generates nerve signals through the autonomic nervous system, particularly the sympathetic nervous system, to the gut, which changes everything from contractions to transit to secretions to permeability to blood flow. So, any gut function can be taken over. So this gives the gut a pretty unique position that all our emotional states that we can, if a good observer can see in facial expressions of a person, are also happening at the gut level. We can't see it there, but we do know that is happening.
What are some examples here? Like if I eat something I know is not going to make me feel good, I eat, I don't know, like a White Castle belly bombers, and I'm like, oh, my stomach's hurting me. What's going on there? Is it interacting with my brain? And why would my stomach hurt if I eat something that doesn't agree with me? And why would I feel like, well?
So there might be 2 components. So if you already know beforehand this doesn't agree with you, even before you eat it, the brain would send signals down to the gut to put it on alert. So it's not the same, it's not the same condition, or I would say, the table is set in a different way than for a celebration. The table in the gut, so to speak. And once the food gets down there, it's likely that your gut will react in a fashion that's not ideal for absorption and transport. If there's something in that food, let's say there's a virus or there's a toxin or something that makes you feel bloated, obviously that's being encoded then in the gut itself, and the gut's nervous system will react to this either in form of trying to get rid of it through diarrhea or through vomiting. But also it will generate signals, and that's the other part of the brain-gut interaction. It will generate signals that go from the gut to the brain. So the brain monitors what goes on in your gut all the time. There's more traffic in your brain-gut system or axis from the gut to the brain than from the brain to the gut. Most of this, the brain keeps outside of conscious awareness, because you would go crazy every time you eat something that you become aware of every single sensation and contraction in the gut. But if it's something that doesn't agree with you, it will send signals. The brain, the warning lights go on, and you will become aware of it. So cramps, gurgling, unpleasant feelings, sensations, pain, all this is transmitted to the brain and becomes conscious if the brain decides this is potentially bad for you.
Okay. What kind of emotions come from the gut? Is it, when you eat certain foods, does that trigger certain emotions to overtake you? Like certain things you eat, you may feel satiated and satisfied and happy. Certain ones, again, you may feel bloated and uncomfortable.
Yeah. So
what kind of emotions go on, and where do you think this comes from?
Yeah. So the basic emotion with food intake is a pleasant emotion. It's satiety, which, goes along most of the time, unless you overeat, goes along with a pleasant feeling of contentment. And, it's not a specific emotion like, happiness, or even though that can happen as well. If you have a meal in the context of a celebration, then this meal will amplify that feeling, if you're in a wedding or birthday party. That's so in terms of the positive sensations, it can also do the opposite. It can create anxiety.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
If it's something, as I mentioned before, that you have been concerned beforehand, before you eat it, that it's going to generate some unpleasant sensations of bloating or gas, then eating this type of food will make you anxious. So these food-related anxieties are very common. So in terms of other depression, that's not really established well, that a particular food will make you depressed. But the opposite is true. If you are depressed and you eat, comfort food, you will feel better. Not a specific emotion. You'll just reduce your negative emotion. You feel less depressed. And that's a sort of a danger of overeating, stress-related eating, that people do feel better from, eating the French fries or the sweets or other snacks, that they're not necessarily good for you. So it's a big modulator, and it's not surprising because there's a lot of cells in your gut. It's not just a tube with smooth muscle that contracts and, generates peristalsis. There's a lot of hormone-containing cells in your gut, particularly in terms of satiety and appetite, hunger, that are releasing these hormones are being released. For example, if your stomach is completely empty, you release a hormone called ghrelin, which makes you feel hungry. After you've eaten, particularly if it's sort of a high-fat, high-protein diet, it will trigger the release of other hormones further downstream in the small intestine, which will tell the brain, you're full and makes you stop eating. So that's kind of the very basic communication. And that communication happens, these hormones are being released locally. So then they act on the vagus nerve, which carries the signal to the brain. But they're also released into your bloodstream and go in a slower fashion to your brain through the systemic circulation.
So what happens if someone's hungry versus hangry? They're really hungry and they're irritable and all that, versus just, I'm hungry, I'd like to go eat. What do you think the difference is? What's going on?
I would say it's a more, being hangry is sort of a more complex emotional feeling. So the basic sensation or the basic feeling would really be being hungry. And if, depending on how you react to this, you would add the anger to it. It's not necessarily connected to it. If you embark on a period of fasting, you're not going to get angry by not responding to the hunger feelings. You sort of overcome that. If you're stressed out and hungry and you don't have access to food, you will, this combination will generate anger plus hunger. Then they interact with each other.
I just didn't know if it was even possible to, on a biological level, quantify what's going on differently between being hungry or, I'm like totally starving, or I'm hangry, or, I'm satiated. You mentioned hormones, but, the action of the bacteria in our guts, has anyone thought of some kind of experimentation to quantify what's going on when we feel these different things, what metabolites are being released, et cetera? Yeah.
So if you bring, so far we've talked about the brain-gut axis by itself. When we bring the microbes in, obviously it gets more complex and less well understood. I think we pretty much know most of the things that happen between the brain and the gut by itself. But then you bring in 40 to 100 trillion microbes that all produce their own chemicals, these metabolites. And these microbes are also in part of this bidirectional dialogue. So if you're stressed or angry or depressed or sad, the signals that go down to the gut and also affect the microbes, either directly, because some of these neurotransmitters and some of these hormones that are being released from the gut can act on receptors of the microbes, changing the behavior of the microbes and what they then generate from the food that you eat. And on the other hand, there's so many of these chemicals that the microbes produce, hundreds of thousands metabolites, that some of them act on the gut itself and on the enteric nervous system. Some act on different fiber types of the vagus nerve, signaling to the brain, and some get into the bloodstream and through the blood-brain barrier and affect the brain directly. And then on top of all of this, you have the interactions of the microbes with the gut-based immune system, which is just microns away from the microbes. And that triggers a whole nother communication channel of immune molecules that goes to the brain. So if you ask me sort of a simple question, do we know when you're not eating and you're hungry and you get angry, what role the microbes play in this? The only thing I can say is what we do know is if you are fasting for a long enough period of time, that this will definitely affect your microbes in a significant way. They will move away from the further away from the immune system in the gut. They produce less of these chemicals like short chain fatty acids or all the other metabolites. And so there's definitely, if you fast, it will affect the microbes in a way that they then contribute in a different way to this communication between the gut and the brain. But as a specific answer to your question, I think we're pretty far away from that. It's just it's too complex a system. This is like, I always use this term. It's a supercomputer that's very finely tuned, and we're just trying to understand what individual chips in this supercomputer do, but we don't really understand the entire system.
Another question. So, who eats first? Does that even make sense? Do bacteria eat first when food gets to the stomach, or do we eat first? Or is it a passing back and forth of my cells take a whack at digesting this, this food I've eaten, then they hand it off to the bacteria and they go strip other stuff off the carcass, and then it gets passed back to me. And what do you imagine it looks like, the dynamic?
Yeah, this is a great question. So, and then this has to do with where do the microbes live? So there's definitely a gradient from the stomach downstream all the way to the end of the large intestine, that the density and the richness and the diversity of microbes increases the further you go down. There's, we all know there's one microbe in the stomach, quite well known, H. pylori. But then you go down further down, first part of the duodenum and the jejunum, a very low density of microbes. So, but if you go down into the end of the small intestine and the colon, you get the biggest density anywhere in the world of microbes in one place. So that influences what happens to the food that you ingest. So the food is broken down initially mechanically and chemically in the stomach, then in the first part of the small intestine. Most of it is absorbed right there in the duodenum, the very first part of your small intestine. So depending on what you eat, if you eat food that can all be broken down and absorbed in the small intestine, then you feed yourself first. And that would be, for example, so with the typical standard American diet, most of it would be absorbed in that very beginning of the small intestine, because everything is processed, very little fiber, it's ultra processed. But if you eat a diet like our ancestors used to eat, that is, much, much higher in fiber and plant-based food components, there's a lot of these very large molecules called polyphenols in plant-based food that cannot be absorbed in the small intestine and that have to travel down. So both the fiber and these polyphenol molecules all travel down unabsorbed until they get to the microbes, and then the microbes begin to break them down into smaller pieces that are being absorbed. So depending on your diet, you are being fed first with the typical Western diet. And that's one of the reasons why, we have our obesity problem, because it's very easily absorbable, high caloric density. And if you eat a largely plant-based diet, like our ancestors used to eat, then the process is very different. A much smaller proportion is absorbed proximally, and most of it happens really at the end of small intestine, large intestine region. And it's one of the reasons why, when people ask me, what is the healthiest diet? And there's sort of a culture war going on, as between the vegans and the keto defenders. And I would say it's actually quite easy. If you if you pick a diet that feeds primarily your gut microbiome with these large molecules, with low caloric density, then you're doing the best for yourself as well. So this means you want to have a diet where the microbes come first, and then our nutrients and our well-being and our human absorption early on comes second. So it's the most fundamental question, really, in terms of evaluating what's healthy for us. And we have deviated in developed societies quite a bit from that principle.
Is there such a thing as bacterial economics? I made this up. So I was thinking, if I'm a bacteria in someone's gut and I produce a short chain fatty acid in exchange for like a certain sugar, what's the economics of that? Do I want 3 molecules of sugar and I'll produce 1 of short chain fatty acid A? Or is it 1-to-1 or 5-to-1? And what determines that? And what do you think those ratios are? And could we discover them?
So, I mean, this term bacterial or microbial economics is actually a good expression, because I always say that like short chain fatty acids are kind of the currency that microbes use to interact with us, with the host, with our gut, with our health. And, short chain fatty acids are basically what microbes produce from when you feed them complex carbohydrates, like, fiber molecules. Let's just stick with fiber molecules or these polyphenols. So the fiber molecules are broken down by an ecosystem of collaborating microbes. So it's not every microbe does the same work in this process. There's some that come early on that break down one chemical link, and then that modified carbohydrate molecule is then processed by the next group of microbes. So it's an ecosystem, or like a factory with different components in it. And ultimately what comes out are these short chain fatty acids that have a beneficial effect for, mainly for us. I mean, the microbes themselves derive energy from this metabolism, but ultimately they use this currency to pay us. And you could sort of take this further in a simplistic way. There's clearly a symbiotic relationship between our microbes and our gut. The microbes produce stuff that is beneficial for us, and we, or our gut, delivers services such as a stable internal environment, a safe environment, and a constant supply of food. So I think the economy, you could say, is the microbes pay for these services with the short chain fatty acids that they produce, and we, or our gut, pays them back with these services. That's probably started from the beginning, when in evolution, the first microbes, by coincidence, decided to settle in the gut of the most primitive marine animals. So there's this concept of an economy is right on, to the way you could see this. It's rarely presented like that, but I think it's a very good way of saying it.
And then in food, I've only seen one study about this, but I don't really know if it's conclusive. What happens to the DNA and RNA inside the food we eat? Do you think that we make use of it somehow? Or is it all just burned up in the stomach? So
that's a good question. I mean, there is a phenomenon called lateral gene transfer, that, for example, microbes can transfer, or have transferred in evolution, some of their own DNA onto human cells in the gut. That's probably how our neurotransmitters in the enteric nervous system and ultimately in our brain came about from molecules that the microbes had developed. From the food itself, I don't think that there's any transfer of that genetic information, because it would be an extremely dangerous design principle if that could happen. There are organisms like viruses and like microbes that can do this in principle, transfer, genetic information. But there's no way that food could do this, because, all the different kinds of foods that we eat with different genetic material, it wouldn't make any sense anyway. If that exchange happened between or happens between, dietary components or plant components or animal components and microorganisms, that's another story. I mean, there's definitely a communication and exchange of design principles, because some of the plants have similar molecules, like similar hormones, estrogen-like compounds that we find also in the microbes, but also in our human system. So there's clearly that link between plants and food and microorganisms, but not directly to us humans.
I don't know. I mean, what happens if I eat fermented foods and it has bacteria all over it and it survives the stomach acid and those end up populating my gut? Or if I take a probiotic that's designed to survive the stomach acid, I mean, that's not natural, let's say, but I mean, wholesale bacteria and probably associated phages get into our guts. I mean, they end up in our body. I wonder if some of them stay in residence so they can, maybe the RNA and DNA in the foods we eat aren't actively changing us, but maybe our bacteria can, maybe some are left by the time they get to our colon bacteria, and the bacteria can either incorporate elements of it they find or not. Maybe they use them to alter their interaction with us. I don't know.
Yeah, so this, what I said just a minute ago, this principle of lateral gene transfer is definitely something that is known, that can happen between cells, certainly between microbes. So microbes in evolution probably have done this on a regular basis to transfer genes from one type of microbe to another. And also, as I said, I think that gene transfer probably almost certainly happened between certain microbes and human cells, but particularly, in terms of transferring synthetic pathways or transferring neurotransmitters, that's almost certainly the case. Also, we know our mitochondria are actually, originated as bacteria that have been incorporated as an intact, microbial organism into our mammalian cells. So the transfer, not only in terms of genetic material, but in terms of the entire microbe. So there's clearly this flexibility. But your question, if DNA from the plants can go directly to our human cells and being absorbed and influence us.
This is probably gross, but if I think about the colon, it's in different states all the time. It's empty. It's then filling up and stool is forming, and then there may be quite a bit, and the person goes to the bathroom, and now it's empty again. So its environment would change, I would think, very dramatically in this cyclical way. What do you think that tells us? Does that reveal any mechanisms about what's going on? Has anyone kind of looked and sampled, someone's stool, let's say, on a longitudinal basis, and seen how it interacts with, what it tells them about what the composition of the bacteria is in the colon, over time?
Yeah. So first of all, this gut microbial system is not static. It changes not only with the diurnal rhythm. So it's different when we sleep and when we don't eat for 8 hours. Definitely different. The microbial interactions with our gut cells, our immune cells. Also after a bowel movement, where we get rid of a very large number. I forgot what the percentage is. I mean, could even be 90% of the bacteria transiently, leave the colon. The thing is, there's a blueprint that exists in our gut that even if, there's a marked decrease in the abundance, it will come back exactly with the same composition and the same players. It's so fluctuations in total amount or, density does not influence the stability of the basic blueprint of your gut microbial ecosystem. So there have been studies, during what happens after fasting. Most of these studies have been done in mouse models, and they have generated a lot of interest in terms of this intermittent fasting, where you can manipulate the population and the interactions of the microbes with our gut. And so I forgot. So your main question was, how much do we know about this? Or
well, that particular environment, like the sigmoid colon, the end of the line, again, if I was a cell living there, my environment would change a lot. Like, again, it would be empty at one point after a person goes to the bathroom. Then it starts filling up and stool starts forming in there, and it gets to be pretty large. The whole area is filled up. Now I'm in a totally different environment. The stool is full of bacteria, and there's materials in there that are used or not used. Now the person empties their colon again. Now I'm back in this old environment. And so it's like a very, I don't know, it feels like a very dynamic environment to be there. So has anyone studied the dynamics of the lower colon, the sigmoid colon? Because again, I think you'd see a huge changeover of, materials and chemicals present, bacteria present, etc.
Yeah, that's, it's a little bit more difficult to study this in humans, because if you want to look at the stool and regional changes of the microbial ecosystem, which means in different parts of the intestine, that's quite difficult to do in humans. Also, typically people have a bowel movement once a day at a particular time, so you can't really say, I'm going to compare this to what it was like right after the meal or right after a bowel movement. So this is not easy to study. There's, it's definitely something that the field will move from looking at stool samples that are taken, they're not even always taken at the same time. When we do studies, we don't tell the study subject, I need a stool sample exactly from 6 o'clock in the morning when you wake up, and I need another one after you've eaten. It's in humans, it's just not, you just can't do that, and so we rely on mouse models. And as I said, the mouse models have been studied with.
It would be a good model for this is rabbits. My daughter's had rabbits, and those things eat and poop constantly all day long. They would be probably really easy to get that done because they're just constantly doing it. Yeah, no,
but then it would be hard to compare this to the human GI tract, which functions quite differently. That's always a dilemma, to extrapolate findings that you have in animal models who have different patterns of digestion and defecation, and then, make a conclusion what happens in humans, because in humans, it's affected by your emotion, how well you slept, what you ate the night before. So, some of the questions, this is an excellent question. I would say, what we can say, there's a lot of fluctuations, tremendous fluctuations throughout the day, also throughout the night. So, for example, when you're fasting, your whole contractile pattern in the GI tract changes to one that's a rhythmic pattern. So every 90 minutes, you have a big contractile wave starting in the esophagus, going down all the way into the colon and moving everything with it through this wave. So, the microbial environment changes dramatically every 90 minutes when you fast, which happens in most people only during sleep. So that's another variation that comes in, but overall, it's clearly a design of a system that can deal with tremendous variations, but remain stable. You don't change your average diversity or your average richness or abundance, relative abundances of the individual players, which is remarkable. It goes through dramatic changes throughout day and night, but the system stays the same. And that's kind of when I said earlier, that the expression, the brain-gut microbiome system is the best expression, because systems are designed in engineering to have both flexibility and stability, tremendous stability, resilience, resistance to change. So I would say the best answer to your question is this, what you're asking is exactly what the brain-gut microbiome system does all the time, maintaining stability in the face of tremendous variations.
When people talk about diversity, they harp on about diversity. What do they mean? Do they mean strain diversity, metagenomics diversity? What kind of diversity means something when you talk about the gut?
Yeah, certainly strain diversity is an important one. So, many take the earlier techniques like the 16S RNA, ribosomal RNA techniques of measuring relative abundances didn't really get down to the strain level. Stayed at, at best at the species level, most typically even at a higher category of classification. But what's really most interesting is the diversity at the strain level, at the finest granularity. And so diversity just means, how many different strains are there? So you could say you have a diverse ecosystem, but your different strains are represented by a very small number. Extrapolating this to a city where you say it's a highly diverse city, this could mean, you have one African American family, you have one Native American family, you have one Korean family, and you have, many families that are Caucasian. So that would be a diverse population. What's important, but that wouldn't really be a good ecosystem with resilience and resistance. What you would need is richness of these individual players. So the combination of richness and diversity is really what determines the stability of an ecosystem, if that makes sense to you. So
okay. I guess I figured as like a job center, there's multiple different bacteria can do the same job. And, in our guts, there's certain, there's calls for certain things to be made and certain things to be produced. And depending on what you are, you either can work in the job center or not. But again, you're not the only one that can work there.
Yeah, that's true. But so you could say, you could have a diversity based on white collar and blue collar workers. That wouldn't tell you too much about the actual property of that ecosystem. If you break these white collar and blue collar workers down into the subcategories with the greatest granularity, in a society like the US, you would have a tremendous, variety and diversity of the type of people that do specific things and are very specialized on. Just look at medicine. In earlier ages, you would have internists and surgeons. Now you go into internists and you break it down into not only into cardiologists and pulmonologists and gastroenterologists, but each one of those subspecialties has, again, several subspecialties. So the more diverse that system gets, the more connections are between these individual players. The expert who does the endoscopies on your gut needs to communicate with the expert that does with a pathologist and with a nutritionist, and so it forms a network. The key here is the diversity creates networks of players, and the networks are between what we call in network science the edges between hubs and between nodes. It's the number of edges that connect, for example, one node that makes, for example, one node a hub will have much more influence. And it's very similar. And we do apply the same kind of network mathematics to the microbiome system. And I would say, to answer your question, the diversity is one criteria that gets you to a very complex network. And it's the network stability that we're interested in.
Okay. If I shrunk you, into a tiny size and put you in my large intestine, what would you observe? Would you see spider webs of biofilms kind of overlapping each other and interacting with each other? Or what do you think it looks like? It's not just individual bacteria just, floating around in there like a soup. I would think it's structured somehow, right?
Yeah, you would actually see and experience a lot. So first of all, you would need an oxygen tank because it's an oxygen-free environment. You would need lights because it's completely dark. So, you need the oxygen, you need the lights. Then what you would see is a very complex, structured geographical system. It's not just everything floating around there. We know this, for example, from the microbiome in your oral cavity, in your mouth, where spatial structures have been identified. So the network is not something that is flexible and floats around. It's really structures between the individual microbes that create an architecture of the system. So you would see that. You would see, a big mucus layer that is like a big fence that separates you that are swimming in the middle of this from actually getting close to the wall of the gut with the immune cell sensors. And you would also see that, there's different communities. So the way, if you slowly could go down from your small intestine to the colon, you would encounter totally different communities with different players, different environmental conditions like the pH, the acidity. So it would be a fascinating journey through your gut, not just seeing the big things, but also this whole microbial world, not just going like with a submarine through it, but it would be with a microscope that allows you to see all these individual details of the system. It would be fascinating, to see that.
Yeah, it's like that movie from, I think, the 60s or 70s, like The Incredible Journey, where they shrunk down these people and injected them into this guy and inside this tiny little submarine. I don't know
Exactly.
if you ever saw it.
Yeah, but in the 60s, a lot of things we know now that are going on in the gut, they didn't even know, so that journey, if you made this movie today, it would look very different. It would.
I wish they would. That'd be super cool if they made a movie like that today.
Yeah, no, I think it'd be great educational tool.
Yeah. Well, very good. Well, Emeran, let's restate where people can get your book. I guess it's on Amazon and Kindle and everywhere. Is it published yet, or what's the date?
It's coming out in 4 weeks. Okay. Yeah, you can go to Amazon, or if you go to my website, it lists all the booksellers. So I encourage, and the publisher encourages people to go to a diverse group of booksellers, not exclusively to Amazon. You can pre-order it now. And there's if you look at my newsletter, there's certain prizes that we give for people that do that. So from free copies of my first book, signed copies, to access to a 1-to-1 or 5-to-1 conversation with me, answering questions. So I would like to encourage people to order this book now. And
the book is called The Gut Immune Connection: Understanding Why We're Sick Can Help Us Regain Our Health. And I'm speaking to Dr. Emeran Mayer. His name is spelled E-M-E-R-A-N. Last name is M-A-Y-E-R. So Emeran, thank you very much for coming.
Thanks for giving me this opportunity to talk to you.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.